Okay, we're live.
Yes.
2 minutes, 2 more minutes.
Then I will start recording. Okay. Shall I put my headphones on or can you hear me?
Yes, I can hear you. No problem. If you want, you can put your headphone. People are joining right now. 71 people are there. I hope every participants got our registered participants got our study materials, and maybe after this webinar we can available for everybody, because we don't know that how many people are interested on that webinar, but materials are available on our website, because speakers made these things for everyone, so we have no problem to distribute it. But till now, we provided all the materials, study materials for the all registered participants. And if time permits, we allow few people to join in Zoom directly for the interaction session. Those are very new to R. Please carefully learn and see the whole webinar. I'm sure today you will give some confidence to how R is working or not. Yes, I got many questions, many requests that how to download it and how to install it. So please figure out these things from the YouTube, or I request to, our speakers to little bit, I give them idea, though it is link is available where it's need to be downloaded from the study materials. So as you all know that it is a very important nowadays to learn this type of language. And in Bioingene we try to figure out that bring everything for the our viewers and also the community, students community, scholars community. Our next webinar in the plant science, which registration is ongoing. June is already decided. In July, we are bringing 2-3 webinars on intellectual property right, patenting, and this type of things. So I'm now in middle of the discussion with the speakers. So in July, we are bringing such type of 2-3 webinars related. And July also we bring a Women's in Scientist series webinar on the funding and fellowship related talks. So time to time visit bioingene.com to registration process. Please forward information to your colleague and friends, because in Bioingene community, our aim is to make a bigger community. Everything is free here. So don't worry about charges and all. Everything is free in Bioingene. Soma, I request, it's now time, it's just 1 minute past. Go for the introductions. Go for the introductions of Bioingene, and that in between I show the website where the informations are put into the webinar related, where the certificates is available or not. Then introduce our speakers. In between, I will send the Zoom link to the participants after 30-40 minutes or 1 hour. Let's see. Okay, Shoma, go for it.
Okay. Hello, everyone. Welcome to another Bioingene webinar. Bioingene is a non-profit organization created to promote plant science research worldwide. The webinar series has been designed to build a platform from where plant scientists can present their research to the world. We hope future plant scientists can gain perspective and inspiration by listening to the esteemed plant researchers talk about their scientific accomplishments and their thoughts on building the future of plant sciences. We are grateful that many renowned scientists have accepted our invitation to share their research insights with us. We are also thankful to you, the viewers of our Bioingene family, for your interest in Bioingene webinars, your constant support and appreciation. We have received a huge response through our registrations for today's webinar. You can register for the webinars through our Bioingene website and social media pages. You can live view all our webinars or watch a recording later on our YouTube channel. Please subscribe so that you don't miss out on any of our live events. For attending today's talk, you can apply for after attending today's talk, you can apply for a certificate of participation via the feedback link, which will be provided after the presentation. Today's session is also special as you can interact with our speaker and talk about today's topic, that is R and data visualization. Please type in your questions related to today's talk in the YouTube chat box for the Q&A session. The topic for today's webinar is an introduction to R and data visualization by our guest speaker, Dr. Vishwanathan Satish, scientist, Genome Informatics Facility, Iowa State University, USA. Dr. Satish is a research scientist at the Genome Informatics Facility at Iowa State University. His journey into the world of molecular biology, biotechnology, and bioinformatics began at Tamil Nadu Agricultural University in Coimbatore, India, where he earned both bachelor's and master's degree in plant breeding and genetics. He then continued his education with a PhD in molecular biology and biotechnology at ICAR, Indian Agricultural Research Institute, New Delhi. As a research associate at ICAR, National Institute for Plant Biotechnology, he worked on a bioinformatics project that required identifying genes involved in adventive embryony in gugul, a medicinal plant. Concurrently, he began studying phosphate starvation responses in plants through various collaborations. One such collaboration led him to work as a visiting researcher at Nanjing Agricultural University in China, where he deepened his understanding of plant stress biology, particularly phosphate starvation responses and their signaling mechanisms. Shortly after, he assumed the role of a postdoc fellow and later a research assistant professor at the CAS Center for Excellence in Molecular Plant Science in Shanghai, China. In these positions, he functioned as both a biologist and a bioinformatician. He has recently moved to Iowa State University, where he analyzes next-generation sequencing data and develops analysis pipelines and also creates data analysis tutorials and workshops for sciences in the United States Department of Agriculture. We are highly honored to have him on Bioingene. Dr. Satish, this virtual platform is all yours now. Dr. Satish, you can share your screen now and we can start.
Okay. Okay, sure. So thank you, Shivo and Soma, for the kind introduction and also for the invitation, onto the platform to reach out to several of our, your viewers and students across the world. So before we start, just a quick look at what we're doing here at the Genome Informatics facility at Iowa State University. So we tend to the needs of several scientists within the university, outside of the university, in industry, and from the government on analyzing data, mostly biological data information that we have that comes in, like it's being generated at a very fast pace. And one of the key things, which is more relevant to our topic today, is that we are working on tutorials, so that can be used, which is open and can be used by people all over the world. And so this is just the web page where we are creating workbooks for data science, bioinformatics, and geospatial analysis. So the geospatial analysis workbook is currently, we have just started working on it, so it's still incomplete. But something more mature is the bioinformatics workbook, which we began in the beginning, and like about 10 years ago, around 2011. And then currently we are working on the data science workbook, which is near completion. So taking a look at the bioinformatics workbook, so this is how, the layout of the pages. And you have different topics that you can sort of browse through. For instance, the project management, and then the bioinformatics terminology that you might want to use, you might want to know about and start using them once you get in the field. Experimental design, which is very important, because, for example, you may, there was once a time, when a lot of experiments were done without any replicates. For example, RNA-seq, people just, sequence it once in one sample once, and they say, let's try to identify something from this. So those are certain wrong, experimental designs. So we talk a little bit about that here. And then, a simple, introduction to BLAST, what it is, and so on. It's just a tutorial that you might want to go through if you are interested in. And then just taking a look at what RNA-seq data analysis holds. So it just gives you a complete pipeline of what we can do when we work on RNA-seq analysis. So if we have a reference genome, how can we do it? So it gives you the different experimental design. You have the data sets that are available, which you can actually download from the EMBL. Okay, so you have the code which is used, and you can try this on, and if you have the right kind of software for it, and also the platform, like, for instance, Linux or Mac or Windows, because now Windows also supports Linux, and so on. So you go through, how you download the reads, then how do you check the quality of the reads, and so on. So you could, go through these tutorials and try to learn some of these analysis yourself. So the next one is the data science workbook. We have several aspects to it, and I think even if you were to start with bioinformatics workbook, the one place you need to start is the command line. So this basically tells you a lot about what a terminal is, how to, enter commands into your command line, and how you can sort of use that to, do some analysis. You could go in there, and something more relevant to our discussion today is the programming intro. And for instance, introduction to R programming. And you have, this is basically the material that we are going to do today, though it's organized a little bit different. And you have, the next step into that is using a package called dplyr in R, which is used for advanced, data manipulation and so on. So you sort of have that. And you also have the plotting package, which we'll be covering today. So talk about a lot of, aspects of data plotting, how you, read data in, and then how you separate them variables and so on. And then another important aspect of data analysis, exploratory data analysis, is that's how you start, when you start analyzing data. That is, you go through it, you want to understand what the data is, and this particular package sort of helps you do that. Okay. And so this sort of, tells you, create some good graphics on what the data looks like and so on, and what information you can derive from the data. So all this is, based on R, the package R, which is an important statistical package that is very useful for biologists or for any field, if you're interested in analyzing data. So I think that is a quick introduction to what we do here at the Genome Informatics facility. So I think now it's time to just move on, into our subject proper. So so I did get a request to just show, students how to download and install R. So I'm on a Mac machine. So this is my page. This is the page. You can just search for R download and Mac. That's the first hit that you get on Google. So you go into it, and then you have this package. So you could just click on it, and that gets saved And maybe I'll sort of save it to my thing. And then if I were to run it again, so it asks you, like, okay, so this is a different architecture. So I need to sort of download a different package. So for example, this one, because the architecture is different. And I could just try to open it. Okay, so all you need to do is follow, because R is already installed on my system, and I'm not going to go through the different steps, but this is how we get started with installing the software. Okay, and all you need to do is just follow through. And in the end, what you get is an icon, which says R, and then you could sort of, like double click it to open it. But the thing is, we are not going to use this base package for our session today. What we need is something called RStudio. So RStudio is, okay, sorry. So RStudio, so you need to go to this down, a particular page, then download RStudio desktop. For, in my case, it's Mac OS. And if you're on a Windows, it should show you Windows and directly download it. So this is another one. And you can also see that this is, the thing is already been downloaded. The RStudio is already been downloaded here. This particular version is, so that's why you get one over there. Okay. So you could just download it and double click it. Okay. And then, just follow through, like the tab that appears, you can just follow through and then install it. All you need to do is at the end, you get a symbol. I'm just going to cancel this. So you're going to get something like this, a studio. And you get like that. So, okay. So this is the RStudio thing. So one of the important things you need to understand here is that you need to have your R, the base R package, installed if you need R to work. So with that, okay, that's a very quick, because I was not really, thinking about showing how to do the installation, because I was expecting that most of the software would be easy to install, but I didn't think it would be a bit of a problem here. But then this is the interface that you will get once you sort of open the RStudio package. Okay, so with that, I think we can now move on to the actual presentation. So this is the interface. So this is called an integrated development environment. So where you can sort of type your code in, like, for example, this is the console. This is called the console window. You have 4 windows here. You have the console window. This is the environment window. This is the, the script window. And this is where you have all the plots, it shows up the files you have, and so on. The different packages that you might have installed or not installed, and so on. So, for example, in this case, you have a check mark here, so saying that all the base package is already installed here. Okay, and probably loaded. I'm sorry. So this is the different libraries that are already installed, but these are the ones that are loaded. Okay. And this is the help. So for example, you have a data set that's called Faithful, and it's one of the data sets that we're going to look at while doing the plotting session. And you could sort of, go through this particular help page to see what the data is all about. Okay, and so on. So let's go to the console window. So this is where you sort of, explore, and you just try to type in your commands and see what happens. So here you can see that I'm on R version 4.2.1. Okay, And so this is called a prompt. This symbol right here is called a prompt. So this is basically to tell you that this is where you need to enter your command.
Satish, excuse me.
Yeah.
I think your screen is not shared, which you have talked about. Actually, we
Oh,
are.
okay. Great. Sorry.
Okay.
Oh, I'm sorry. Let me just.
Yes, now it's okay.
Okay, so I need to go back again and
Two, three slides back.
yeah. So so once you open this RStudio, so I'm going to just go back and to retrace my steps here. So this is like the RStudio interface, and what you have on the left. So you have 4 panes here. So what you have on the left here, showing this where I'm highlighting. So this is called the prompt, the console, where you enter the commands like so. No, sorry. So you can see, like already you're running into some errors. So we'll see what these errors are as we go on. So 1 + 1 is 2, and so on. So this is where you enter the commands, and then we have the environment. We'll see what it is, as we progress. So this is where we have the script window where you could sort of enter commands. For instance, 1 + 1, and you can sort of run it from here, and you can see that it is run on the console on the top. Okay, so I can just say, press Control L there so that I can clear the screen over there, and then go down to the script window and press this run button. And you can see that it is executed on the console. So, and then we have the this particular pane where we can see the plots. We have not created any plot yet, the different files in the particular folder. The help where you have information, like regarding, for example, in this particular case, it's the faithful data set. And if you want to understand what the data set is all about, you go to the help page and so on. So now what I'm going to do is I'm going to show you how to create a project, so that we can have all the information related to our project in one place. Okay. So what I'm going to do is I'm going to go right here to the file, and then I'm going to say new project. Okay, so it takes a bit. Okay, so you have this pop-up window saying that, okay, let's start a new directory. Okay, we're going to create a new directory here, and then we're going to start a new project. The directory name, I'm just going to say Bioingene workshop. Okay, so what am I going to create this directory? It's going to be a directory is just basically a folder. So it's going to be on the desktop. Okay, so I just click create project. Okay, so now we have the project open. Okay, so you can see this here, the Bioingene_workshop.r project, so which will contain all the information within this particular project. And in fact, if you want to open it up, you can just directly click on it and then it will open. So you have the name of the project right here on the right, on the top right. Okay, so let's just see what's there on the okay, so I think, okay, I'm going to stop sharing here for a bit here. Maybe I should be sharing the entire screen or the entire, like, yeah. So you should be able to see the, yeah, so that, and yeah, so you should be able to see the different files on the desktop. And Okay, so we have two folders on the desktop. One that you can see is this Bioingene workshop. And then it has got only this .r project file in there. And then we have another folder, which is files, and we have certain files. Okay, so these are the files that we're going to use in this particular workshop. So what I'm going to do is I'm going to copy them. Okay, and then I'm going to transfer them into the Bioingene workshop folder, as it were. So you can see that we have that in. And now when I open up the RStudio window, so you can see that these files are in your particular folder. So now I think we are ready to get into, what we're going to do next. That is load a particular file called the .rmd file. So this is called an R Markdown file. Let me just type it in here. It's called R Markdown. So basically, it's a format. Let's open it up. I'm just going to click on it. Okay. So when I click on it, you will see, and this window is adjustable. So I'm just going to pull it up a little bit. Okay. So you can see that it has got a certain format. You don't have to worry about what's on the top. This is about, different information like title, who created it, and the output format if you were to sort of create it into a PDF or a HTML output or whatever. Okay, so basically what you need to know here is that we can have text. Okay, so this is where we, type in text to say that, okay, for in this case, you can go down to this website to download and install R and so on. And then we have another set where you have something like this. Okay, so this is called a code chunk. So this is where we enter the code. So what is the advantage of this is, so I'm going to open up another thing. So this is 3 tick marks, the backticks. These are called backticks. And then I'm going to type in R, and then I'm going to sort of end it with this. So this is going to be a code block. And within this, I can do something like this. Okay. And then I can go up here and you have this run button, just click it and then say run selected line. And you can see that you have the output right here in the file itself. Okay. So this is a great way to learn R. So you can see that the code is also executed on the console, but it doesn't matter. So you can just make it full screen like this, so that you can just concentrate on what is happening within this R Markdown file. So this is the platform that we are going to use to learn R today. So just to give you a heads up, so our idea today is to sort of if you will, is to create this particular, figure here, okay, a plot. So how do we do this? But this is going to be a long-winded approach to doing it because you're getting started. You need to know the basics of the language, and how to create a particular data set, and how to read data set in, and then perform actions and perform computations on those data sets that you might load and you want to understand a little bit more about. So now that we have seen that, okay, how to enter the code in the R Markdown, here you don't have to do anything. If you are following along, you just need to run the code. That's it. So for instance, here in this case, we have just one line of code on line 35. So it's not going to be 35 in your R Markdown because I've created this. So you can just follow it on, with reference to what I'm saying on my system here. So let's take a look at the basic operations. So our idea now is to build a data set. Okay, so we sometimes what we have is we have data sets that that are generated, or you get it from a friend, or you have developed it on your own, probably on Excel sheet, and you want to read it in and analyze it. Yeah, that is one way of, like reading data in it. The other way is like you can try to create a data, and there are several times where you will have to create, you will be creating data sets within your own system. So in this approach, what we are going to do is we're going to first try to build a data set. Okay, let's see how a data set looks like. And to that, like we start with the very basics, and then we just start building it. Okay. So I've shown you this already. So 1 + 1, and then we can do the additions. So this is line 43, and then I can just still run. And this is, you can see that the cursor is on line 43, and then I can go here and I'll click run. So you can see that it's 2 - 1 is 1. Yes. The other way, the keyboard shortcut to this is, you could, if you're on a Windows machine, it could be Control and Enter. Okay, you should press Control and Enter at the same time. Okay, so now I'm pressing Control. Even on a Mac, it works. It could be Command Enter in the Mac, or even Control Enter, both works. So pressing Control and Enter, you have the output again. So it's the same. You don't see any change there. 2 × 2. So this is your operator. Okay. Operate for multiplication, and then you do have what do you call the a division symbol there with the /. So that's going to be division, and then we run into the exponential. So how to create an exponential? So 2 ways of doing that. Okay, so with a double asterisk and outside with the cat symbol. Okay, so these are the ways that you can sort of run commands on the R Markdown file. Okay, so what if I wanted to run this entire chunk? Let's say this is all a contiguous flow of commands, and I want to run them together. So for that, what we do is you have an option here, says run current chunk. So that's Shift Command Enter or Shift Control Enter on Windows. And if you were to run it, you can see that the entire code block or the code chunk is executed with the the answer displayed below it. Okay, so this is how you work with the R Markdown file, and then you can interlace it with text. So this is, nothing happens here. Okay, this is just pure text. Okay, now not only numbers, you can even represent letters. Like, for example, within double quotes you can have A. Even with single quotes you can do that, and you can say A. Or you can have a string of letters, which is a word, and then a string of words. Okay, something like this. And like I said, so here I'm doing, just for the sake of Windows users, Shift Control Enter or Shift Command Enter for Mac, the entire code chunk. Okay, so this is the output that we get for this particular code chunk. Okay, now, so this is all fine, like you want to perform computations. And sometimes these are very simple calculations, but there are some certain kind of times when you need to remember a lot of things. For example, in this particular example, we have a number which is large enough, which it's difficult to keep remembering. So what do we do in such contexts? So one thing that we can do is we can create what is called a variable or an object. So you can liken it to a basket. So in this case, the symbol that you find here, like the less than symbol followed by the hyphen or the dash, it just says we have this number on the right. You need to take it and assign it to the object name on the left. So basically, this is the the assignment operator. You can also use the equal to, but the convention is you go with a less than and a hyphen. Okay, I'm sorry. A less than and a hyphen to show that it is an assignment operator. Okay, so what happens is when you assign it, the the value is assigned to this particular object. So I'm going to run this particular line 61, control enter. Okay, it runs. And then what you see on the right here is a value gets created, A. So this is the environment, and this is the object that we have created right now, and this is in memory. So this is stored to the memory. Okay, and it holds the value, this particularly large value. And now we're going to go down here and execute the line 62. So that would be, this number. And you can see that this particular object is also created in the environment. So I'm going to run the next line, 63. So A, so you can see that the number is displayed. So, and then we have 64, that is B. And if you were to run the entire code chunk, and this is the output you get. Okay, now we have a good system to hold large numbers. And can we perform operations just using this, variables like A and B and so on? Yes, we can. So in this case, I'm just going to run the whole chunk there. So you can see that we get the output. What's A plus B? You minus B, you multiply it, you go for an exponentiation. So you can see that it reaches infinity on line, the output of line 72. Okay. So can we do this for characters? Can we have a single character or a string of characters? Can we set them to be variables or objects? Yes, we can. So this is the output that we get. I'm running the entire code chunk here, So here you can see that we are writing the word language, and we are assigning it to the variable name X. And then we have R is my favorite programming language, and we are assigning it to the variable Y. And then we are displaying the contents of the two variables. So this is the output that we get. So another way of, printing the output is also using the function print. So print X and print Y. And if you were to run it, the whole chunk, you get both. Okay, so these are different ways. So now we have started building, like a few things, we saw what a number, scalar is. We have now been introduced to objects. We've been introduced to variables. Both are the same, very similar meaning. And then how to sort of print them onto the output and so on. So now we come on to the next part of our, the tutorial, and that is what is a vector, an array of numbers. So how to create this array of numbers. So there are multiple ways of doing it. We're going to see two of them. So one is using the C function. So just basically means concatenate. That is, you've taken a list of numbers, a range of numbers, and then you put them together. And the other one is using the seek function. We're going to see how to create numbers with both this, functions. So in the first case, we have C. So the syntax is you have C followed by the 10 numbers. Okay. And that goes within the parentheses. So 1, 2, 3, and you clearly know what is going to come out of this. So I'm just going to run that. So I've executed line 91 with control enter or command enter, whatever your bit on your operating system. And then on line 92, we have Y using the seek, SEQ function to create the numbers. So that's within parentheses 11,20. So I'm going to run it, but we don't know what it holds, right? So I'm going to execute now line number 93, which is X, and X holds a list, a string of numbers starting from 1 through 10. Okay, so let's see what Y holds. So Y seems to be holding numbers from 11 through 20. So this is, like you can see the difference here already. So there are two different ways. There are other ways, but then it's two different ways to get started with. to create a list of numbers. Okay, so now can we perform mathematical operations? Because the key thing is about having vectors is to be able to perform computations on them. So can we perform computations? So x plus y. So we have two objects here, or variable names here, containing certain numbers. So let's try to see what happens if we were to add them together, x and y. So what we get is 12, 14, 16, and so on. So basically, 1 plus 11 is 12. So what we see here is element-wise, addition of the numbers. So what happens if we are multiplying it? So that's x into y. So that's going to 1 into 11, 2 into 12, and so on. And that is the output here. So again, it's going to be element-wise. For matrix multiplication, it's going to be different, but we are not going to look at that today because this is just going to be a very introductory session. Okay, so you can try for the other operators and see for yourselves like how they behave. Okay, so the next thing is, okay, we have a vector, like 1, 2, 3, and so on. And how do we extract? Like, for instance, you want the fourth element in the vector. Okay, like we have the vector x, and say we need the fourth vector. What do we do? So that is where indexing comes into place. Like, okay, so these are contained in an M. So each element in the vector has a position. Okay, and that is called index, and it starts with 1 in R. In certain programming languages, the numbering starts at 0, but in the case of R, it starts with 1, 2, 3, and so on. So here in this case, how do we, for example, I'm just going to execute our x. So these are the numbers. So what if I want to have the first number? Okay. So I'm using the square bracket notation to access the first number in the vector x. So when I do that, you can see that I get the number 1. What if I want to get the number, the fourth element in the vector? And you can see that I have 4. What if I want to get the 11th element? Okay. So here you can see that you have an NA, which stands for not available. And that is obvious because we only have 10 numbers here. We don't have an 11th, 11th element in this particular vector. So that should make sense. Okay, so we are getting the first element out there. Now, so are there any ways, like let's say we need a range of numbers. Okay, how do we extract them? Okay, so here in this case, we are going to use the y vector. And here you can see the syntax 1:4. So let's see what this yields. So executing line 115, we get the output 11, 12, 13, 14. So this basically means that get me the numbers that are indexed at positions 1, 2, 3, and 4. So 1 through 4. And those are the numbers that we get here. So let's say we need alternate numbers in the vector. For instance, in this case, we want 1, the first element, the third element, and the fifth element. So to retrieve those elements, what we do is we use the function C to concatenate them and then run it. So you can see that we get 11, 13, and 15. So the numbers 12 and 14 are not returned. Okay, now let's say we don't need those numbers in these particular positions, like 1, 3, and 5th positions. So what we do is this, we could say that I don't need it. So a minus symbol there, a -1 or a -1 or a -3 and a -5, concatenate it, and what you get is 12. So you don't have the element on in the first position, that is 11, and the third, which is 13, and 15, which is in the 5th position. And the other numbers are returned. So the next thing is like, okay, so you can see that the minus symbol is sort of, common there. So you can just bring that out. You could say minus C, and then the positions that you don't need, 1, 3, and 5. And then you can just run it, and you can see that you sort of, let's say we put 6 here. Okay, we don't know. So 15 should be there, and 16 is missing. Okay, so this is how we access different numbers or different elements in a given vector. We have a few elements, a certain number of elements in a given vector, and then this is how you sort of pull them out. Okay, so the next thing that we need to understand is data types. So these are very important, particularly when you do analysis. And those are integers, numeric, logical, and characters. So how do we represent them? Okay, so the numeric, in this case, I think you're getting the point here. Like we have a number 1, and then we are assigning it to the variable name n. So just will represent the numeric. So here in this case, you can see that I have something like this. Okay, so when you have a hash symbol, or in some cases it's called the pound symbol, so it means it's a comment. So it means it tells the program that this is a comment and it should not be executed. So let's run this. Okay, so when I run it, you can see that the object is created, n is created, and it is and the value that is assigned to n is 1. Now, if I were to remove the hash symbol, now this doesn't have it, and you can see that already we have an X mark over there. So that is one of the advantages of using RStudio. And it says unexpected token numeric. So something is gone missing. Now, what happens if I were to run it by mistake? So you get an error. It says unexpected symbol and then sign like equals 1 numeric. So now you go back there, add the hash symbol there, and you see that the check mark over there has disappeared. And now when you run it, you can see that you don't see any errors there. Okay, see the other one, the next data type is the integer. So the integer is represented by 1 and then followed by a capital L. It's not really necessary to do this, but then when you use this notation, it just says that, okay, you are, assigning this to be a integer. Okay, but it's not, we have other ways to just make integers. But this is the representation that we have in R. So then we have logicals. So logicals could be true or false, and then we can sort of assign it to L, and then we have characters, and the fourth one is the character, some string. And then let's just execute that enter code chunk. So you can see that N, L, I, and C, they're all created. Okay, nice. So now, so like I said, these are variables, and they are sort of, it could be scalars if they are just one element in them, or it could be a vector if there are multiple elements. So how can we identify or count the number of elements that are present in each of these, say, for example, vector? So that we get by using the length function. So the length of n is going to be 1. So it's all going to be 1 because we have just 1L, true, and some string. So even though this is some string, it's together. It's one, entity. So you can see that they're all of length 1. So each one has got one element, and each in this case, just single element. So it's got to be scalars, but they have one element each. Okay. So then we use the class. So this is the next function, to understand what class it is. And then using this symbol on the different objects that we have created, we have numeric, integer, logical, and then character. So this is how we identify, if you are running an analysis, you've created a variable and you're not sure what its type is and the data type is. So you use the class symbol to identify that. Okay, So now, so far we've seen, we've created variables with a single element. Now let's create a vector with different. So for numeric, integer, logical, and character vectors. So here in this case, what I'm doing is concatenating three numbers, 3, 3.0, and 5. 5.0. So those are floating point numbers, and it's also called a double in R. And you have the integer vector, so given by 1L, 3L, and 5L. The logical vector is going to be true, false, and true. And the character vectors is going to be is going to be the first element, A is going to be the second element, and string is going to be the third element. So we have 4 vectors, and each vector has got 3 elements. Okay, so I'm going to run the entire code chunk. They are all created. Nothing is displayed below because we are not printing them out. Okay, so now let's get their class. So you can see a new kind of syntax here. Okay, so we have class, and we know what this does. Then we have a semicolon. Okay, and then we have something else called length. So we know what this does, the length of the numeric vector. So that is supposed to give us the length, the number of elements in the numeric vector. The class of the numeric vector should give us the class. But what is this syntax? Now let's run it and see. Okay, so you can see that numeric. So the class of the numeric vector is numeric, and then the length of the numeric vector is 3. So this is a new syntax so that you can have multiple commands on the same line, and you can separate it with a semicolon. Now, if I were to remove this and run it, you can see that there is an error. Okay, so once you put that back, it works. So the other thing is you can still run them on separate lines. That is also possible. And you could run this line with or without a semicolon. It doesn't make any difference. Okay. So generally the convention is like you can use them, but then generally nobody uses it. So that is the syntax over here. So let's check the class and the lengths of the different vectors. So now we have a vector here, like numeric of different data types. So now you can see where we're going with this, like in the sense that we are trying to build something, with just a few letters here and there. Okay, so the next step is like, can we create vectors and then put them together? And to be able to do this, what we're going to do is we are going to create something called a matrix. Okay, so the first vector we're going to create is V1, and that is going to be the numbers 1 through 5. V2 is going to be the numbers 6 to 10, and V3 is going to be the numbers 11 to 15. So I'm going to run the entire code chunk. So you can see that the 3 vectors V1, V2, V3 are created. So V1 is 1, 2, 3, 4, 5. V2 is 6, 7, 6 to 10, and 11 to 15. Now, how do we put them into a, column, arrange them column wise? And to do that, what we're going to do is we're going to bind them by column. Okay, so the C here represents column bind. Okay, we're going to column bind the 3 vectors. So the 3 vectors being V1, V2, and V3. So now if I run it, so we have this, we have this particular output. Okay, so now you can see that we have built a sort of a data set, but then still, there are a few things that we still need to learn. Okay, we have 3 vectors, but then when you put them together, we see that it's turning out to be a data set here, okay, carrying certain values, each vector carrying certain values. So what we can do is we can write that, this entire output into an object like before, like you would do for a vector or a single scalar value. So you just run it, and I'm going to run the entire code or code chunk and see my mat is going to display this particular matrix. So let's check the class of the matrix. Okay, the matrix is going to be matrix or an array. Okay, so there's another way of creating a matrix. That is, if you have a set of numbers, then you can sort of use what is called the matrix function. Okay, so in this case, we have matrix, and then we have certain values called arguments. So here you have 4 arguments. So that is, the first argument is a set of numbers that you want to create a matrix with. Then you have the number of rows that you want in the matrix. Then you have the number of columns that you want in the matrix, and then how you want to, enter the numbers in the matrix file. Okay, so how do I know how to use this particular function, matrix? Okay, to know that, we can take the help, of this particular function called the question mark. Okay, so this is a function that is available, and then when you add a question mark before that, it will open up. So for example, in this particular field, you have the help, and you will see that it will open up that particular tab to tell us what it is. Okay, so you can see here that we have matrices. It's a matrix. It's from the base package. We'll read about packages as we go on. So what it does is it creates a matrix from the given set of values. Okay, so that is what it does, and it creates a set of values. And this is the syntax. So the data, the first argument. So these are called arguments. Okay, so this is the data is the first argument. NA just means there's nothing. We need to provide it as a certain set of variables or something like that. Then the number of rows, the second argument is going to be number of rows. The third argument is going to be the number of columns. And here, in this case, it says by row equals false. So what it means is the numbers are going to be added column-wise and not row-wise. So we're going to take a quick look at how that works. So I'm going to, go back here. I'm going to do the same thing, matrix 1 to 20. So this is the data, okay? The first argument is data, okay? And then we have the number of rows. That's going to be 5, okay? And the number of call is going to be 4, okay? So I'm not going to add the by row equals true, okay, argument yet. So when I execute this particular function, you can see this output. Okay, so what are the total number of rows we need is 5. So 1, 2, 3, 4, and 5. So we have 5 rows, 4 columns, and that is what we need. And you can see that the numbers are entered by column 1, 2, 3, 4, 5. That's down the column. Then it starts at the top 6, 7, 8, 9, 10 on the second column, and so on. And you are also noticing this particular syntax here, which means 1 comma just means whenever you have this particular notation, it just means you are on the first row, which means when you have a comma 1, you have what you call the first column. Okay, so if you were to execute, take this particular number, the way we represent it is 1 comma and then 1. So that means 1 comma 1 just means first row, first column, and that is the value. Okay, so now I'm just going to add by row is true. Okay, and when I run it, you can see that now the numbers are entered row wise, like 1, 2, 3, 4 across the row, then 5, 6, 7, 8 across the second row, and so on. So this is the difference. So, okay. And we run it. So this is the output that we get. Okay. So now we have seen a couple of ways to create a matrix. But then when you think about a data set, it does not just have numbers. It can have, logical vectors, like it can have characters. You can have factors, like, for example, male, female, that will be entered in one of your columns. So how do you account for that? Okay, because one of the problems is matrices is that it can hold only a specific type of specific data type. And in this case, if it is numeric, all the values should be numeric. If it is character, all the values should be character, and so on. So let's see what happens, okay? So I think you remember that we have already created these vectors. They're already created, but then what we're going to do is we're going to put them all into a matrix, okay? cbind. So looking at this particular function here, like expression, is that we are going to column bind the numeric vector, integer, logical, and character vectors, okay? And then assign it to the new mat or the new matrix variable name. And then I'm going to execute the entire code chunk. Now you see that there is something funny happening here. And you can see that the numeric vector, which should be a number, it's showing as a character, okay? In fact, everything has been, kind of coerced into a character. Everything has become a character. True is not true. This is a character. It's not a logical. To use true or false as a logical, it should be without quotes and in the uppercase notation. So I am the character it is. So this just shows that the character, if you have a character vector in your data set, everything gets converted. So now to sort of circumvent this, like, let's see what the new matrix says. So it is still a matrix. So everything is converted into a particular data type, okay? So this is where we are going to use another data structure called the data frame, okay? So let's see. So now we are going to do what we're going to do. We are going to build a data structure called data frame. It's going to house certain variables. So we're going to sort of use an example from plant biology, and then create 4 vectors, and then we create a data frame, and then we see how we write that data frame that we create into a file. Okay, so that is what we're going to do in the next steps. So here you see that I'm using a function called set.seed1234. And that is because I'm going to use a function called rnorm, which it means random normal. So I'm going to create a data set which contains 100 values, and it has got a mean of 110 and a standard deviation of 10. So that is going to be the plant height. Okay, the plant height, and I'm assuming, and in this case, I'm just saying it's just going to be like a rice plant, and it's going to be 110 centimeters tall, and the standard deviation is going to be about 10. Okay, so it's going to be about 100 different plants. So what I can do is I can just going to select this, and I'm not going to, when I'm executing this, so I'm just doing Control Enter. And you can see that you have 100 values generated there. And you can notice the first value, it's 121, okay, and 20. So I'm going to run this function again, okay, and this is going to run it again. And you will see that this value has changed, to 85. I'm going to run it again, 125. So that is why it is a random normal, generator, value generator. So to avoid this, what we do is we set a seed. So 1, 2, 3, 4 is the seed number. I execute line 221. Okay. And then now when I run it, I get 97.92. Okay. So now if I run it again, it will change. But then if I want the same value that I got before, I run the seed again and then run this step again. So you can see that it's the same value that I'm getting here. So this is just basically to reproduce what I'm getting here. You use the seed, okay? So now what we're doing is we set the seed. We are setting the seed. We're creating the plant height, and we are just there are 100 values. We don't want to look at all the 100 values. We just wanted to look at the top 6 of them. And for that, we use the function called the head. Okay. So when you do head and plant height, it gives you the top 6 values. So 97.9234, 112, and so on. You know? Okay, fine. So one thing that you notice here is, we have too many decimals, so we can sort of round it off to 2. And for that, what we are going to do is we are going to use the function round. Okay. And then the number of places is going to be 2. Okay. Like I can just run this code. I'm going to select this code and just do Control Enter. And then you can see that they're all, like 2 decimal points now. Okay. So I run this entire code chunk. Okay. We have created plant height. Okay. So now the next thing that we are going to do is we are going to take some flowering data. Again, it's going to be random normal, but here you can see I'm nesting the function. So this is called nesting of functions. So the first thing is you're creating a random normal value of 100, but then you have multiple, decimals. So you just run the entire thing here. Okay, so you just have a whole an integer number, because flowering cannot, you cannot say 105.1 days and so on. So we just round it off to the nearest whole number. Okay, so we run the entire code. The flowering data is ready, and you can see. So you should sort of use the same to get the same seed set.seed command and the number to get the same values that I get. Okay, so now the next thing that we do is we run create the spikelet fertility, and then we have the new function that we are using here is max. Okay, in this case, max means the maximum value of spikelet fertility. So we have 100 values and we are displaying the top 6 with the like the head command. If you just want to see the bottom half, you just use tail. Okay, the tail just gives you the bottom half of the vector, and then this gives you the maximum value of the vector. So that is 95.27. And then we also create for seed set. Okay, great. Now we are going to create a data frame here by combining them, like column wise. So we have seed-bind, plant height, flowering 50, spikelet fertility, and 1000 seed weight. And I'm going to execute line 256 there. Okay, and then I'm going to take a look at the top 6 rows. So you can see, row 1, 2, 3, 4, 5, and 6. And you can see the values here. Plant height is here, flowering 50, spikelet fertility, and 1000 seed weight. Okay, so let's take a look at the class. So it's going to be a matrix because they're all the same type. Okay, so it's going to be a matrix. And particularly when you're using the function cbind, it is going to be a matrix. Okay, so now what we do is we have all these values, but we don't have IDs, the ID, the plant ID. So we need to create them. And the way I want to do it is I can just go on to the way I want to do it is like 1 to 100. Okay, but then the last number is 3 digits, so I would like to do something like this: 1, 2, so on, up to 100, just to make sure that we have 3 digits. So how do we do this? To do this, we use something called paste, the function paste. And here what we're going to do is we're going to paste 2 zeros, and we are going to add them to 1 through 9. Okay, so this 1 through 9 just going to create numbers 1, 2, 3, 4, and up to 9. And if those are there, and it's going to add 2 zeros before these numbers. Okay, so when I execute this, you can see that we are getting something. Okay, but then we have a space there in between. We do not need that. So what we do is we have another function called paste 0. Okay, and this by default there is no space. It just puts them together. Now, if you were to use the paste function alone, then we can use something called the separator argument and just leave it with two double quotes. So that would be a space. This would be no space. And when you run it, you can sort of, create these numbers. And this is what we're going to do here. Okay, we are going to create a vector A1. And in this case, it's going to be paste 0, 0, 1 through 9. The separator is going to be nothing. Okay, no space. So you run that. And the next thing is we are going to go from 10 to 99. And we're going to add a 0. And in this case, we are using the function paste 0. So basically, we just run it there. So if I just select A1 and run it, you can see, okay, it is 001, 002. Okay, that's fine. And A2 is going to be, okay, so that's great. And now we are going to call this genotypes. That's a variable name I'm going to give. I'm going to create a vector of this. And that's going to be, it's a vector. So you can see it's just C. Okay, I'm then putting them all together. C, A1, A2, and then the last number that is missing is 100. I secure it, and then I run the genotype. So we have 001 going through to 100. So we have created the variables now, the genotype IDs now. So now the next thing is, how do we bind them to the data that we have? So we already have that as my data. We have put the plant height and the other variables, the biometrical traits, into my data. And now we're going to combine them and then run it. So yes, but the problem is, like we saw before, everything has been converted into a character because the genotypes is a character. Okay, so here you can see that they're all within quotes, and that means that's a character. Now that is converting everything else into a character. So we cannot use this particular function cbind. So what we're going to do is we're going to use the function data.frame. We're going to put the genotypes in front so that that becomes the first column, and the rest of the data here in my data goes as the second, third, fourth, and fifth columns. So I'm going to run the entire code chunk here, and now we have a working data set. Okay, so we have genotypes, we have plant height, flowering 50, spikelet fertility. Just expand it. I can just move the screen a little bit, okay, so that we can see it completely. And this is what we have. Okay, so and let's check the class. The class is going to be field and the data frame. Okay, we have another function in R, which is called stir or str, which is basically structure of the data. And when you run it, it gives you a good overview of what the data is all about. So it's a data frame. It's got 100 observations, like 100 values. So each row is an observation, and each column is a variable or an entry there. And so we have 5 variables, and the variables are going to be genotype, plant height, flowering 50, spikelet fertility, 1000 seed weight. So you can see that genotypes is character and the rest are numeric. Okay, now we have a working data set that we can perform actions on. Okay, so now that we have this data set, how do we write it? Okay, to what do you call it, to your disk. So we use the function write.csv. There are multiple ways of doing that. I'm just going to show you what write.csv does. So here what we do is we create the object name, because that is the object we have created here. Okay, we have called it field data. Okay, and that's the data name. And we are going to give it a name. Field data, comma CSV. CSV means comma separated value. We'll see what it is. And once we have saved the file. And the quote is equal to false and row.names is equal to false. So these are 2 options that we need to use, because there are certain values that will be given in quotes which we do not need. And then the row name, sometimes when you, like for example, in this case, the 1, 2, 3, 4, and so on, that will be considered the row names, and that will be returned in the data set. We want to avoid that. And therefore we use row.names as false. So if I just run this code, we don't see anything, because when you go to the files tab, you see that the data is being created. Now I'm just going to click on that and view it. So that opens up a new window there, a tab there on the console, or the script window, I mean. And then you can see that they are separated by a comma. So that is why this data set is called a comma separated value. You can even have tab, the data points separated by tab and so on. So genotypes, comma, plant height, comma, and so on. So every value, genotype 001, comma, 97.93 is going to be the plant height, then comma, the next value, comma, the next value, and so on. So this is a a data set that we have created. So now I think it sort of gives you an idea of what R is all about. Okay, we understand how to use R. This is the most important step. And what we have seen up to this point is like the core elements that we, that we need to know if you want to get into data analysis. Okay, so now what we're going to do is, so once we have analyzed data, the way we want to describe them is by making plots. To understand them, we need to make plots, and that is going to be the second session of our tutorial. And in this case, what we're going to do is we are going to use a system called ggplot2. And this does not come along with the base R package installation. It is a separate package that you need to install. So we have more than 10,000 packages, 16,000 probably now. So you need to install, and to access some of the functions within these packages, you need to install them. Okay, so you read a little bit about what ggplot2 is here, in the short introduction given here. But before we start doing anything, what we'll do is we'll just go on to this particular line. Okay, so that's called 337 install.packages. Okay, so you can just delete it. If you're working along with me, you can just delete that particular hashtag. So remove the comment. So, and then run it, okay? Because I'm not going to run it because the GG2 plot is already installed on my system. So, even if I did run it. So, yeah, okay. At the moment, I cannot, because there are multiple packages that need to be. So you could try installing it on your own. And because we need to use that package for plotting, okay? So I'm going to comment this out, okay, so that it doesn't get executed. Okay, once you've installed the software, we need to load it, to make sure that those the functions that are available within ggplot2 package, we need to load the package, and that we do by using the function library. Okay, so I'm going to do that. Okay, so now it is loaded. So so what is this package? Okay, so it is based on a certain grammar of graphics that was written by Leland Wilkinson. Okay, it gives a theoretical foundation for producing graphics. Okay, so we have a book on it, and this is the link to that particular book if you're interested. And then this was made use of by Hadley Wickham, who wrote this particular package. He is the author of several packages on R-based packages. And in this particular tutorial, what we are going to do is we're going to use two data sets, Faithful and Iris, which are two data sets that are found within the base package, to explore ggplot2 a little bit before we go on and try to do something that we promised to do in the beginning of the workshop. Okay, so here what we need to do is we need to keep a few things in mind. One is the data. The data is the most important aspect. Without the data, there is no visualization. Okay, the mapping. Okay, so like when we created the data set, we had the genotypes, we had the plant height, and so on. So each variable there is important, and that needs to be mapped with the data. Like, okay, we want to say, for example, create a histogram of the plant height. So the data, the variable plant height should be mapped to the histogram. So how do we do the mapping? And that is important, okay? And then there is a certain kind called facet mapping, which means that we can separate the plot that we create based on the variable names. So we are going to see what these are. It may not make sense, much sense now, but as we go on, we'll see what they are. And then the geometries. Geometries is basically a histogram, a scatter plot, and so on. So that those are represented by a geom underscore, and the function is given by geom underscore followed by histogram or point or a bar for a bar graph and so on. And there are certain things that the the background and the layout of the plot, and then certain scales, like for example, color scales that we can use to make the figures more interesting. Okay, so now that we have loaded the the package called ggplot, let's take a look at the data set first. Okay, so I've told you what the str function is. It's the structure function, gives you a glimpse of what the data is. So there are 272 observations and 2 variables, and the 2 variables are eruptions and weighting. So eruptions is the number of eruptions and the waiting time between these eruptions in minutes. This is given in the waiting is in minutes. Okay, so let's take a look at the top 6 rows. You can see the top 6 rows of the data set, eruptions and waiting. Okay, the 79 minutes, 54 minutes, and so on. Okay, so the first thing is the data. What we need to do is just plot the data, and that is given by this particular command called ggplot. Okay, so that sort of creates the layer there. So what we're going to do is we are going to build this data layer by layer. So this is going to be the first layer, and the first layer is going to be data and faithful. So I'm going to say the argument name is going to be data, and the name that and the object that I'm going to use is faithful. So when I run it, you can see that it creates a canvas, but there's nothing there. Okay, so the next thing that we do is we map it, like what do we want to do? For example, we are going to create a histogram, and for that we need to create a histogram of the eruptions. For instance, in this case, we do that by saying, okay, the data is going to be faithful, and we are going to set a mapping. And the mapping here is the AES stands for aesthetics. So we are setting up the aesthetics that we want to work with. And then it's given by X equals eruptions. So eruptions is what we have here. Okay, so this is the column that we're going to plot. So when you run this, so you have a slightly different layout, like different grids. You can see different grids, but still nothing is getting plotted there. Okay, just the layout and then the grids. So you can see that the first layer, and on top of that, we are having a second layer that is being added. And now what we're going to do is we're going to say what needs to be added to that as a third layer. And in this case, it's going to be a histogram. So the way we do that is that, okay, we put in the information that we need to involve there, like these, okay? And then we need to add this plus symbol over there. So you're going to the next layer. So you're going to add the next layer, and that is going to be the geom histogram. And when I run it, so we have a histogram here. So nice. So you can see, it can give you a little bit, means the idea of how it runs. So you can see that the clear, 2 very clear distributions. Okay. So there's another way we can do it. One is like giving all the information about the data and the mapping with the ggplot command here, right here. And the other way of doing that is you can also give that entire information within the geom histogram function call. So when you're adding the layer, so here you can see that you don't have any information here, but all of that information that we put in here goes down to the geom histogram, and it's still. So this is another way of doing it, but it's always better to use it this way, like where we have the entire data and the mapping given here. So you might be seeing that, okay, here I did mention the data and so on, but here in this case, I'm not mentioning it. I'm not saying data equals faithful and mapping equals, because that is the order in which the variables appear for this particular, the default, positions where each of the data sets are. So now if I were to have something like this, now in this case, I need to mention it. Mapping is this, and the data is going to be faithful. Because if I don't do this, then it's going to get confused. Because what ggplot expects is that the first argument is going to be the data. The second argument is going to be the mapping. So now if I run it, you can see it just gives you this plot. But if I were to just remove that, it still does. Okay, and that is because I've already given the the data option here. Okay, because now if I remove this, yeah, so you can see that you run into an error here. Okay, so I think you get the idea there. So in this case, what we're going to do is we're going to put the faithful in the beginning. So that is the data, and the second is going to be the options. So here you can see we are taking the faithful data set. We are going to create a histogram of the eruptions, and we are adding a few more details in here. So we are saying black color. We are saying that the color is going to be black and the fill is going to be white. Okay, let's see what it does. And then we are using a theme called classic. Okay, so let's see what happens there. Okay, so take a look at what we get here. We have grids and a different kind of a layout. And here you can see all those grids are gone. We have a clear, X and Y axis lines. And then, like the color is black. So which means the lines are in dark black, and the fill, what goes within the bars, is white. Okay, so that is what we find here. And what is this theme? So this theme classic is this particular layer that we have. Okay, so now what I'm going to do is I'm going to comment this out, and then I'm going to include this line. Theme is going to be BW. So like I said, we need to add a layer. So then so this is the second layer that the second layer we're adding, and this is the third layer we are adding. But in this case, we are commenting it out, which means that when you run the command, it's not going to be taken into consideration. Okay, so now if you were to sort of remove this, okay, let's just see what the theme BW does, and then we'll show you a few things more. So this is the black one. I can say this is a different format that you have. So now I'm going to comment this out, and then you include this, and this will be the minimal, the theme. Okay, so now if you remove the plus symbol there and sort of run it, you can see that it goes back to the original layer that we have already. Okay, so this sort of tells you what we can do with themes, like we are creating themes here, and whichever one that is very suitable for you to present your data. Okay, so color-based mapping. So how do you map? So we want to include colors, like on the graphs. So how do we do that? So here, okay, I think you understand what line 393 does. We have the data set, we have the mapping, that is eruptions. So we are going to create a histogram. Here we are creating another aesthetic, and that is the color of the eruptions. And wherever the eruptions is less than 3.1, it's going to be giving a different color. So let's see what it does. And we're using the theme classic. Okay, so we can see that it automatically, and it sort of gives you a color that it chooses on its own. And everything that is less than 3.1, so you can see the values over here. Anything less than 3.1, we are going to have a different color. Have that map, it's a different color. And the fill, the bar fill is going to be white. And then we also have a legend. Okay, so eruptions less than 3.1. So this is going to be false, and this is true. So true just basically means, is it less than 3.1? Yes, and those are the ones that we see on the left of the plot. Okay. So another thing is we can also sort of fill it based on the mappings. So here in this case, here you can see we set the color, and the color is basically for the bars on the outlines of the bars. But this is the fill, okay? And you can see that we have the bars filled with the different colors there, okay? And then you have a black, the color, and we have changed the colors here. So in the sense that the color is going to be the outlines of the bars, okay? That's in black. So this is how you sort of, create your plot layer by layer. So the next thing is we'll see how to create a scatter plot. Okay, and for that, we are going to use another data. It's called the iris. It's just basically a data frame of 150 observations, the 5 variables. And it's got, the variables are sepal length and width, petal length and width, and the different species. There are about 3 species, seedosa, versicolor, and what do you call it? And just 1 second, let's see what we have there. So I'm just going to say levels, iris, species. Okay. So there are 3 different levels. So here you can see iris species has got seedosa. The 3 species are seedosa, versicolor, and Bioingene. Okay. So this is how you sort of access the different, parts of your data frame. Okay. So now taking a look at the, what do you call the top 6 rows, and this sort of gives you an idea. Sepal length, width, petal length, and width in the species. Okay. So now what we're going to do is we are going to create a basic scatter plot. And I think, if you look at the syntax, you will understand what is going on here. So we are calling the ggplot function. We are providing the data set that is iris that is available within our, you don't have to load it from anywhere. The mapping is going to be the x-axis is going to be petal width, and the y-axis is going to be petal length. Okay, so here you don't have to access the data frames in a different way. And then geom point, and basically what we are saying is geom point means, so geom histogram gives you, histogram geom point gives you a what do you call the gives you the scatter plot. And in this, the aesthetics is going to be, the coloring is going to be based on the species. So we have 3 different species, and we're going to color the points based on the species. And running it, we get something like this. Okay, so it picks up a color on its own, like default colors. Now we can change the color that we use here, and we can sort of, we have a range of colors that we can choose from, and that is given by a package called R Color Brewer, and so on. So this is the range that we can choose from. And in this case, what I'm going to do is I'm going to add a little bit more here, more information here. And this, apart from the coloring, which is going to be based on the species, I can change the size of the dots or the points, the points that we see here. I can change the size of those points. And I can do that by using the function, the argument size equals 3. It's going to be a slightly bigger one. And then the other thing is I'm going to set a different color palette. I don't need this default colors. I want to change it. And then for that, I'm using the function scale color brewer. So this is the scale aspect of the ggplot package. So now the palettes that I'm going to use is set 1. Let's see what set 1 is. So we have set 3, set 2, set 1. So it's kind of this combination. Okay. So when I run it, you can see that I have a separate combination. So all you need to do is just go back. If you think the size is too big or too large, you can do that and you can reduce the size of points of that. Okay. So now faceting. Okay. Now you can see that there are 3 species. Okay. We have setosa, we have versicolor, and virginica. Now, what if we want to separate them, plot them separately on the same plot, but then separately? How do we do that? And that is done by something called faceting. Okay, so how do we do that? And using a particular function called the facet wrap. Okay, so I think you understand what is going on in lines 442 and 443. Basically, you're giving the data and the mapping, and we are setting it to GM point. It's going to be colored based on species. The size is 3, or we can just reduce it to 2. And then we are using it the facet wrap. And here the syntax is use the tilde symbol there. And it's like like this is the function. So based on the species, you need to separate them. And then we are using the scale color brewer. The palette is going to be set 2. So in the previous case, we used set 1. Here we are using set 2. Not necessary, but yeah. So this is how you get it, sort of different, separately. Each variable is shown in a different context. So what if we want to, this is vertical arrangement of the facets. What if you want to have it horizontally arranged? That can be done, just by shifting the position of the tilde symbol. Okay, you can see here, you just mentioned species, see the tilde and the dot, and then you just run it. Okay, so that sort of, gives you enough idea of what you can do with, the ggplot2 package. And this is, okay, so sort of the introduction. So now what we're going to do is we're going to create this mirror plot that, we plan to do in the very beginning of the tutorial. So here, what we're going to do is one thing that we have not done before, and that is how to read an external data set into R. Okay, so in the first introduction part, we created a data set and then we wrote the data into a file within the system. But now what we're going to do is we're going to read a data in. Okay, so the data that we're going to use is the up-down gene numbers. And I'm going to just view this file. So it's just 11 lines. You have the column names, stress, responsiveness, G standing for number of genes. So I have a few stress options as cold, osmotic or osmosis, and then osmotic pressure, stress, heat, salt, and drought. We have genes that are upregulated or downregulated, and then we have certain numbers there. Okay, so this is what we're going to use. And you can see that they are separated, the columns are separated by a comma. So this is a comma separated value file. And to read, so we wrote the file using the function write.csv. But when we read the data in, we use the function read.csv. Okay. And basically what it does is I'm running the entire code there, and you have the data set displayed. So I'm going to give it a variable name, and that's going to be gene nums _nums _mirror. And I'm going to read the data in, and then in line 466, I'm going to display it. So what we have is the stress, responsiveness, and the number of genes. So now what we need to do is we need to create a plot of this, to see actually what is happening. to the plants based on the, the stress. Okay. So we're going to use the ggplot2 package. Okay, so we're not going to change anything there. So what we need to do here is we need to map it in such a way that we need to create bar graphs of the stress based on the numbers there. Okay, so this is what we're going to do. So the ggplot command is what we invoke. We are providing the data set. So that is going to be the gene_nums mirror. So we're going to create a mirror plot. Like, like we have a certain separation, we have a bar graph on the top and a bar graph on the bottom. Okay. So in the aesthetics is going to be, we're going to map the stress to the number of genes based on, whether they are up or down regulated. Okay. Okay. So now when I run it, so let me just run it without any call. So this is going to be nothing. I'm going to comment this out. So that line is not going to get executed. Okay. So I've done it and then say, let me create a geom bar graph. And then so this is going to be a bar graph. And then you run into an error. And this is because what the program is trying to do is to take the numbers, the number of genes, and they're trying to create a count variable or a proportional variable. And that is why it's not giving us what we need. So in this case, what we need to do is we need to add an argument called stat and identity. So use the numbers as numbers. Okay, so we have num genes, don't convert them into some kind of a proportion or something like that. Just take them as numbers and return it. Okay, so now when I run it, you can see, okay, we have a plot. Okay, so, but there is a problem here. You can see that you're not getting it separated into, down-regulated or up-regulated. You can't make it different, but you can definitely see a point over there. A division there, a line there. It shows, okay, it's one thing here and something else on the top. Okay, but this is not what we want. Okay. So the other thing is, one more thing that we can do is let's I'm going to comment this line out. And then I'm going to include this line. And we have stat identity. The position is going to be stack. So stack is putting one on top of the other. So it's going to be still the same. So something very similar to this. Okay. And the width of this particular, the bar is going to be about 0.65. So let me run that now. So now what you're looking at is a different plot, smaller, the width is going to be smaller. But still, we have the same problem, this is not what we want. So let's think about this. So one thing that we can do, so nothing is below 0. So one thing we can do is whatever is down-regulated, we can convert them to like a negative number so that, you have some values that are shown below the 0. Okay, so that is what we're going to do now next. Okay, so this looks really complicated, but let's just, break it down. Okay, what I'm going to do is I'm going to take the data set. And I'm going to access, this is the way you access the, number of genes. And in this case, what I'm going to do is I'm going to take the number of genes and I'm going to access, okay, the number and the responsiveness. Okay. And if this responsiveness is equal to down, okay, what I'm going to do is I'm going to change those values into negative. Okay, so this is one. So this is a little bit complicated here. Okay, so here, and I'm going to access the num genes column. And then again, the num genes, the responsiveness equals. Okay, so what I'm trying to do here is like, typing it down, it sort of helped you break it down a little bit. So I'm going to change the values, wherever we have down, where the responsiveness is down, I'm going to change that value into a negative value. So this is how I do it in R. So you see this call over here, everything, and I'm just taking the ones that are, this is called subsetting. So let me just run this particular code first. Okay, line 483, I'm just going to run it. So it just gives you the numbers, the numbers which are negative, which is down. Okay, so those are the numbers. And then when I run this particular line there, it see that they're all converted into negative. Okay, so that's what I'm just going to do there. So I'll remove it and then run it. So sort of it doesn't show, but when I execute this, you can see it. So you have the responsiveness. If it is up, it is positive, and if it is down, it is negative. So that sort of might help me get along further into the plotting. So now that I have this, I'm just going to keep it simple. I'm going to be creating a bar graph. The stat is going to be identity, and the width of the the bar is going to be about 0.65. And I run it. Yes. So we are doing a little better now. So you can see that we have a set of values that are showing above the 0 and a set of values that are showing below the 0. So minus 2000, minus 1000, 0, and 1000. And on the y-axis, we have the number of genes. And then on the x-axis, we have the different stresses, cold, drought. So it's been, separated. Okay. So now, so let's add a color theme to this, like a theme sort of that is, much more, I think, indicative of what we want to show. So in this case, what I'm going to do is I'm going to add a layer theme classic, and then I'm going to add a y-axis label. So when you look at the y-axis label, you see it's num_genes is just basically, it's just taking this one, the column name. Okay. So I'm going to set up a label for that, and also for the stress, also for the x-axis, which is going to be stress with a capital S. And then I'm going to give a title, GG title, so it basically means that, okay, you can add a title to the entire plot. So these are a few layers I'm going to add now. So when I do that, you have the default, okay, so this is a different layout and different colors, the default colors kick in. And you have a title, on the top. And then you have the y-axis label, which is now different. So one thing that you might have noticed is this legend over here. So we don't have a legend here, but we do have a legend here. So all of this is getting done. You don't have to do anything, but the program figures it out for you. But the one thing that you're different is we would like to have up at the top, on the top, and down below. And here it is basically it's following the nomenclature where you're, it's alphabetical. So down. So you just want this reversed, you want up on the top and down at the bottom. So how do we do that? And that can be done by resetting the levels. So the way we do that is, it's a little bit of, again, some more code. So basically what we're looking at is, we are looking at C up and down, and we are going to set the difference. And the calling us, you can see that there are two functions and three functions that are nested. So you're setting the levels, you're looking at the levels, okay? And the levels in the, the responsiveness column. And then we are going to set the difference based on up or down. And what is going to be presented first is going to be up, okay? And what is going to be presented second is going to be down, okay? So this is going to be the new levels there. Okay. So this is just a prefiguring. I understand this may not make sense, but once you go through the code again multiple times, you will pick it up. Okay. So now we have run that, and then we need to reset the factors. So the factors is going to be, the responsiveness up and down are going to be the two the factors. So we're going to set them as factors. So we say call the factor function, And then gene nums mirror, the responsiveness, and the levels is going to be new levels. So that is what we have set here, the new levels. The new levels is up is going to appear first and then down. So you don't see a lot of change here. Even if you look at them, you wouldn't make it out. Like, for example, you run the gene nums and then you run it. You don't see any change there, but all the leveling, all the re-leveling has already taken place. Okay, now let's run this. So here what I'm doing is, okay, so I'm adding one more level in. Okay, so the we have, yeah, so the title is there, and then we are going to add a few more things here. Oh, okay, so we are on the wrong part. Okay, so here. So here you can see that this gets changed because now we have given a labels here saying that, okay, this can be upregulated, this is going to be downregulated. We are setting the labels for the legend. When you look at the thing above, we have not set any labels for this. This basically takes the value that we have in the data set. Now we are giving it a names of our own, the labels of our own. Okay, so that is additional thing that we have included in this code. And so this is what we have. So this is fine, but okay, there are a few more things that we need to fix. Okay, so the next thing that we are doing is we need to add the numbers here, the number of values. You know, like for example, if it is upregulated, how many numbers of genes are upregulated, how many number of genes that are downregulated. And for that, what we do is we use something called the geome text. Okay, and the aesthetics that we are adding is the number of genes. We need to add those number of genes. So in this case, this is actually commented out. It doesn't run because you do not have a plus there. So when I'm just running it, you can see you have the numbers that are added to the top. Okay, so but the problem here is like, yeah, it is overlapping, and we would like to sort of change it using a vjust. Okay, I'm going to set it to 0 and see what happens. Okay, so it goes on top, you know, on top of the bar, and here again it's on the top of, but what happens if it is 1 and then it's going to be below the line? Yes, it is going to be below the line, but then preferably if this were to be on the top and this were to be on the bottom, it would be great. But with just, okay, even if I use 0.5, which is the default, we get it in between. So here what we do is you use a certain construct called conditionals. So you can see that if the number is positive, okay, we want the v just to be 0. And when the number is negative, we want the v just to be, 1. So that we are using a small construct here, an if-else construct. We are not covered conditionals here, but I think this is slightly advanced, which you may be, you know, you may want to check, you know, once you've gone confident with using R. And so I'm going to comment this out and then run it. And you can see that, you know, the values are displayed on the top and the bottom. Okay, so that is great. Fine. So the next thing is, it still does not cut out to the color scheme that we showed in the beginning, which we wanted to achieve. And before we do that, what we see is that the y-axis sticks is not what we would like. So we want to change that. And so what we do here is we use something called the scale y continuous function. And then we are creating breaks. And the breaks is going to create a sequence of numbers starting from 3000, minus 3000 to 1800. And they are different, and the difference between them, the numbers that are going to be generated is going to be 600. So let's just see what numbers we get here. Okay, so I'm just selecting that number and control enter. And we have 3,000 to 2,400 and so on, up to 1,800. Okay, that's great. And then we're also going to set the labels there. And in this case, what we're going to do is we're going to have the same set of numbers, but then we are only going to take the absolute values. No signs, we don't have a positive or a negative sign. We are just getting the absolute values of that. So that will be the labels. So let's see how the plot comes up and see you can. Okay, so you sort of see this. Okay, we have the 0 starting at 2,400 up to 1,800 and so on. Okay, so now the final part of the code, and that is when you are changing what you call the color of it. And then here we are using another package. It's called Wes Anderson, and you could install the package using this particular by commenting out this particular what do you call line, and then you load the package here. Okay, so you remember this particular last line. So this is when we created the y-axis sticks, and finally we go on to the skill fill manual. Okay, so yours here, the values, the color that you're going to use is going to be the function. This function comes from the Wes Anderson package, and then you're going to use 2 colors, and the name is going to be Grand Budapest. So that is different color schemes that we have here. So let's take a quick look at what we have. What the, for example, I'm going to create a Wes out. Okay, so no documentation, that's because, okay, so that is because I have not loaded the package. So now if I load it, then I run this palette, so you have this. Okay, so we have something like this, and it tells us, there are different color schemes that we can use, like Bottle Rocket 1, Bottle Rocket 2, and so on. So Grand Budapest and 1 and 2, there are different color schemes. So we are using one of those color schemes. Okay. So here we are using Grand Budapest 2, and then we run it, and then, so this is the final sort of plot that we wanted to arrive at. So I understand. So this sort of ends our session today. And I understand it was a very high level, of introduction. So you started with how to use R, the different data types, the important data types, how to make a data set, how to sort of, interact with the data set, then how you read the data set and do some plotting using certain packages that we learned to sort of. So this is actually a material that I generally cover in about 2 to 3 days. So this is, I understand, but the advantage here is that it's all being recorded. So which means that you can always go back to the video and sort of, go through the, and you have the code and the material is already been sent to you. So you have the script with you and you can play with it. You can sort of experiment with whatever you can, and once you understand it, do more. And along as you're working with the script, you can also read more about it. And if you have a problem with any of the concepts that you're not clear about, you just go into Google or just talk with friends and try to understand it. So the idea here was to provide you with something. I understand that this has been a lot today, particularly for the people who are using R for the first time. But then my intention is that with this, you should be able to learn this on your own and sort of use it in your own research. So that is the end of the session today. Thank you, Shuvo. So I'm done. If you, maybe we can take some questions or go into discussion.
Yes, I have collected few questions from the YouTube. And I'm also, Soma will join soon. Soma is there, something. So
Okay.
for your interview session, so I'm going for the questions, 1, 2 questions first. I'm
Okay.
still collecting question.
Okay, sure.
So the first question is, um, uh, what package should use for genetic analysis, calculating dominance, genetic variability, epistatus? Is there any package for like genetics related?
Yeah, there are, like, for example, like when you do GWAS, okay, so we have a thing called GAPIT, which is available in R. So like, but I'm not into that field, but so I'm not able to, like remember any particular packages off the top of my head. But there are several such packages, like that which you can use for genetic analysis. The only thing is you just need to Google it a little bit and see where they are. And most of these packages, they come with a very good documentation. Okay, so there's all you need to do is just install software, go to the on CRAN. So this is called the all the packages are hosted on CRAN, and each package has got a documented in a well written documentation with examples.
Okay.
So what you could do is just install the package and just follow the tutorials or the examples that the packages are giving you. And that sort of, helps you understand how to work with the, what do you call the package? Because, when you're using the package, sometimes you don't really understand in what format the data should be in. Okay, so to understand that, what you need to do is you need to go through the examples. And that sort of gives you the input data, like what the input data should be and so on. And once you get that idea, you make sure that you generate the data that you have, like, for example, from your experimental, output. You prepare the data in that format, and then you read it into the R package. And then within these packages, you have multiple functions which will perform each step that is mentioned in, like a tutorial in every package. And you just need to sort of follow along with that. Yeah. And sometimes what happens is you may run into problems. It could be related to the software itself, like, for example, R, because in a certain, it is the way R behaves, like any program, any software behaves, it will be different on different platforms. So in Windows, you will have a set of, there'll be some errors that will happen, which is very common to different platforms, like operating systems like Mac or Linux and so on. But then there will be something very specific to Windows. There'll be something very specific to Mac and so on. So all you need to do is when you run into errors, just copy the error. Go into Google, paste it into Google, and then search. So you will have, in most cases, what you will do is, what will happen is you will be led into a page on Stack Overflow. Okay, so many of these questions are answered on Stack Overflow. Stack Overflow is a platform where people, programmers, everybody, they try to solve other problems there, collaboratively. So you can sort of see if you have your question answered on the Stack Overflow. You do a quick search. If you think that any of the answers that you provided do not meet your requirements, then you can create a separate question. And like, you can put your error there, and then you can create one. And somebody around the world will probably take a look at it and try to give you a solution there.
Okay. And similar package related questions is there. Is there any package for the phylogenetic analysis or phyllo tree preparation?
Yes, you have. So one thing that we recently, that's a very good one, is ggtree.
Okay.
Okay, so GG just basically means grammar of graphics tree. It's very simple. It's based on the ggplot, package. That's very useful. And then we have others like ape. Okay, I think it is ape. And there are a few other software. So, but ggtree, you can get started with ggtree and see how it works.
Okay. Another question is that as you already discussed, but free data set, is there any source of free data set for the analysis?
Yes, the UC Irvine machine learning. Let me give me a second. Give me a second. Give me a second. Yeah, so it is the UCI machine learning repository.
Okay.
Okay, so you can get started over there. And then you can even, I think you can get data sets on the Kaggle, like there's a Kaggle database. So there again, you have a lot of people, sharing their code and other things. So, and there again, even if they are not hosting their own data sets, they might give you links to, different data sets that are available out there. But you can get started with UCI, machine learning repository. In fact, the iris data set, you can find that even on the UCI machine learning repository. So you can start with there. Once you get into it, then you will know other places where you can find these data sets.
Okay. And similar package related questions, another, but the experiment is different. It's related to stress tolerance studies. So is there any package available for tolerance index, relative stress index, susceptibility indicators, like which generally used in common stress tolerance studies?
Okay, so I'm not aware of any such packages. Maybe they are there out there because, like I said, there are several thousands of packages available. So one thing is, like, when you are writing these indices, so one thing, you can write your own functions.
Okay.
Okay. So for instance, like you have an inbreeding coefficient. Okay. So the formula. What you do is you just implement that in R. Okay. You write your own function. That is so functions is one thing that I did not cover here. Okay. So because one of the advantages of R is that you don't have to learn a lot about programming itself. Basically, you have one line of code. You use a function call and then one line of code, and sort of you get the output. Okay. So for example, if you're using Python, you should sort of know a few things about programming itself.
Okay.
So R is is such a program that you can do statistical analysis, which is well suited for. In fact, R is actually not written by programmers; it was written by statisticians. Okay.
Okay. Okay.
So, so that's why there are certain quirks related to R, and it'll be very different, when you're coming from from learning a different programming language like Python, C, and so on. But then it makes it easy, where you don't have to remember several different aspects of programming languages. You just need to know how to use a certain command. You need to know how to use the arguments within a function. Okay. So these are a few things that you will have to know, and that gets you rolling, and you understand these important aspects that we covered in the first half of the tutorial or the workshop.
Okay.
That is enough to do much more powerful analysis. Yeah.
Okay, so Shofar, we have collected this one. I'm also looking for other questions, and
Yes.
If it is possible for 5 to 10 minutes, we entertain others participants.
Yeah, sure.
For their why they wanted to learn, or what is the necessity to learn, so it can motivate others also, that why they are interested. So we are asking few selected participants, list of the participants for joining our Zoom directly. In the meantime, Soma will do your one interview session, and we are back to 10 minutes after that. Soma,
Okay.
Yes, I am
please
here.
start the interview session.
Yeah. Okay. So it's a wonderful, informative session. And now we are at the point where we will start a small interview or interaction with our speaker. This is a very special section where we get to know more about our speaker, because many of our young, MSc or post, doctoral scholars are looking up to these speakers and how they want to build their career by following their footsteps. So, Dr. Satish, we would love to know how you built your career, and also were there any professional or personal challenges that you had to face while building your scientific career?
Yeah, so challenges are going to be there, for any walk of life. The challenges are going to be there. So in my case, as a, like a school going kid, like probably 10, 11, I knew I was going to end up doing research. So for me, it was more about, like understanding what I would like to do. So back in those days, and even now, like one of the key persons coming from my, like south of India in my state of Tamil Nadu. So one of the icons is Srinivas Ramanujan. So he's one of the all-time greatest mathematicians out there. So everybody wanted to be someone like him. So for me, it was like, should I go into mathematics? Okay, so I started doing a few things as a kid, and then so the question was like, this exactly what I wanted to do. But then, I did not have an answer for that. But why should I go into mathematics? Why can't I go into something else? So by the time I finished school and I wanted to get into college, so because I was going into sort of, I knew I was going to doing research, going into PhD or something like that. So for me was, how do you do research without any bias? So what if I did something that I did not like and see how I liked it? Okay, so that is why I came into biology. So biology is not that I hate it or don't like it, but I was not very comfortable with it. I was more comfortable with, physics or mathematics. So I was in the Tamil Nadu Agriculture University for my undergraduate as well as postgraduate. Then after my post-graduation, it was in plant breeding in genetics. So that was when I found that, there was a huge disconnect between applied research and basic research. And because we are not, like working together, you see that in multiple places, even in China, I was able to see that. But at least in China, they were not afraid to get into a field they were not any used to. They tried to avoid it, but then they were going into it. So for me, it was more about, how do I get into basic research? Because I had the training in plant breeding, conventional plant breeding. But then I thought, if you don't have a mix, if you don't have the ideas of what is happening in the basic research, and how can you implement those things in your, an applied field? And that is a bigger problem. And it's a problem that exists even now. So I wanted to get into more basic stuff, and then I was trying to apply to different places. Something happened. I had to go back home. There was a break of 2 years where I was not doing anything. And not, so it doesn't mean that I was not doing anything. I was doing certain things, but nothing fell in place. So a couple of years later, the break, then a friend of mine once called me and he said, buddy, I'm on my way to New Delhi and just back home for a break. I've booked a ticket for you to New Delhi. Okay. So if you're interested, join me. Okay, because you've already spent too much time not doing anything. So it's time for you to do something. So I booked the tickets, and it's up to you to decide whether you want to go or not. So I decided to go. But once I was in Delhi, I did not get into anywhere because I was mostly, like I said, wanted to get some training in molecular biology. So I could not get into any labs. So about 5-6 months, I was trying different labs. Then I got an opening into one of the labs at IRI. And there I was working for gratis for about 7 months. Then I got into a project where I first started learning basic research, molecular biology. I was working on viruses. Tomato leaf curl virus was the virus that I was working on. And then, unfortunately, the project ended. I had to leave. And then I was applying to other places. But one of the things was that by that time, I was sure I had to do a PhD. And then wherever I was applying, people wanted me to stay in their labs for more than a year, which I couldn't. So I openly told them that that wouldn't be possible. And so for the next 1.5 years, I was not doing anything. So most of this time, what I was doing is I was with friends, working with them on their projects in their labs, like helping them out with their research. And then a 1.5 years later, I got, into my own PhD at the Indian Agriculture Research Institute, 6 years there. So this was the time, sometime in 2011, when I started teaching. Okay, so I think this could be useful information to your viewers, because I wanted to use R, but I did not know, I've absolutely no idea about how to get started. And then at that time, I was creating, I was running a sort of a journal club where I was teaching and conducting a lot of journal discussions, article discussions, and then apart from that, I was also sort of creating inculcating the need for better communication and so on. So then I thought, okay, maybe I can use that platform to teach R. So basically, when I decided that I would teach R, I did not know R. A week later, and I just told them, okay, next week we're going to start with learning R. I had no idea what to do, but just before the start of the program, I knew, I came up with a plan, and that is the same format I follow to this day. But here, it's a little bit more advanced, but then it's the same thing that I've been following for the last 11, 12 years.
So you learn and teach and learn and teach.
Teach and learn. So that's how I learned R, by teaching it to others.
Yeah.
Okay, so after my PhD, RA, and then I was not looking for any scientist position because I was not interested in becoming a PI. And then I was already 38 by then. Okay, so, and I was not eligible for many of the research associate positions and so on. And suddenly they said that, why don't you come to China? Okay. And then that's how I moved to Nanjing. I was there for 3 months, and suddenly, like one of the professors I was working with said, okay, somebody that I know wants a postdoc in Shanghai. Do you want to go? Okay, fine. It was all in phosphate starvation. So then I moved there. We had a good work going on. Unfortunately, 2 years in, we ran out of funds. He could not support me. Then he got some support from outside. So I think you understand where it is going. It's not everywhere I had problems. Okay. And finally, what happened was the center itself got dissolved. And then I had to move out of China.
It's a real roller coaster.
So it's like, that's how research is. So I'll just stop there because, and then currently I'm now in the United States, and I'm expecting more than anything else. So it's not easy, the reason I'm telling you all this is because, if you're in research, if you're not facing these problems, then probably you're not doing research, right? So it happens because, that is the essence of research, because you're trying to find something, and then not everything is going to go your way.
Right.
Yeah.
So how do you envision your work contribution to the broader scientific community and advancing your understanding of plant biology and agricultural sustainability?
So one thing that I decided, when I was in India, is that to stick to agriculture, right? Because one of the problems is that, we have a lot of brilliant people going into other fields, but not agriculture. When I say agriculture, it's not just sticking to agriculture per se, but then different fields of agriculture, So, and that is widespread across plant biology. Okay, we have people that would want to move into other, like animals or so on, but not plant biology. And the other thing is publishing in plant biology is also very difficult.
Yeah,
because I've had friends in China as well as in India, so they wanted to shift, because one of my friends, he became a neuroscientist, and then I had another lab mate who became, who also became a neuroscientist, but his model was in one case it was human, in the other case it was mice. Okay. So if you are getting into plant research, it's going to be very difficult, but then you need to make up your mind that, okay, come what may, I'm going to stay here. Okay. So for me, it's like, it's always, so what the other thing was in my case is like, when I got into my PhD, I knew I would not be able to stay in India for a long time. So what I did was, as soon as I joined my PhD, I started creating a network. So I have a network that is created, and I will continue to work, and particularly with one of my seniors in the Directorate of Onion and Garlic Research, where we have recently come out with a few papers regarding, like how, like transformation, and then we have done, we have already shown how to do CRISPR in onion. And then he's also very interested in creating haploids from any using the Senes3 system. So we are very close to finishing that work, and we are expecting it to be pretty useful. And like considering the fact that onion is very important in the Indian context. Yes. And the problem with onion is that inbreeding depression. So that is one of the aspects we are trying to solve. And the generally, the productivity in India is about 13 tons per hectare. Okay. But if you look at the highest, like for example, Ireland, the productivity is about 83 tons per hectare. So you can see the yield gap over there. Now, how do we, bridge this gap? We are the second largest producers, but then that is because we have a large area under cultivation. And that is not going to sort of, it's not going to continue to be the same way. And like, shrinking land, because of the population explosion and so on and so forth. So that is another thing that, so I'm already, this is just one collaboration that I have. Then I also have collaboration with the senior at the Biotech Research Institute at Raipur, then the NRC Citrus, then in different places, and even in Kerala. So this is my link to, say, India, so that I'm continuing to contribute to Indian agriculture. Okay, so that is something that, but here when you come look at what I'm doing right here in the US, is that I'm, catering to the needs of the scientists in the United States of Department of Agriculture. Okay, so here our job here is to sort of create tutorials and then also to train them in analyzing data. Okay, so here what I'm doing is, when I say agriculture, it is not just the plant agriculture, it's aquaculture. It's like we are assembling genomes of certain species, abalones and so on. So we have a lot of stuff going on. So this is in a way, here what I'm seeing is that I have a wider reach now, not just plants but also different fields of agriculture. So it's very important, like when you are trying to contribute to a certain field, but it also depends on what you're interested in and how you want to sort of contribute. In my case, it's about making things easier for others to do research. So one of the key things now is data science. We have a lot of data, but how do we analyze it? Even expert analysts don't know how to make use of the, like the large amount of data that we have right now. So it's about like how you set yourself forward, and then what you are actually trying to, accomplish. And it need not be something that you accomplishable. It's not. So mostly we are worried about success, but success is not everything. Sometimes you fail, but then, there will be others when that'll be your first step. And then maybe others will come up and then, build on that, stuff. Yeah.
So talking about agriculture and database, what is your opinion on digital agriculture and AI, and how would you combine, what do you think about combining artificial intelligence and agriculture?
It's already there. The minute, the minute you start talking about prediction, it's there. Like drones, drone technology, like plant health. Okay, then estimating yield and so on. It's already there. It's a part of our life now. Only thing, we are not probably exposed to it, but it is already there, and people are making use of it. Right here in Iowa State University, they're using it to understand the crop, diversity, and then even nutrition, the health and general plant health and disease management. So you have people working on it. So it is a reality. It is already there. There's no more going back.
All right. So according to you, which sectors of plant science research do you think will prosper in the near future?
It's about how you apply it. It's not about, okay, so the thing is, okay, you have a problem and you want to solve it. So we cannot just think that, okay, I'm going to solve it like a geneticist would. I'm not going to, it's not going to be like, I'm going to solve it like a physiology. You need to, put them together like a genetics, physiology, or even chemistry, biology, computational biology. You bring them all in, and it's just got to be a multidisciplinary approach to solving problems. You cannot say, okay, I'm a geneticist, I'm going to do only this. And that is okay, fine. But the new, the trend is that, people who are going to be leaders are the ones who are going to be able to sort of spread in different ways, who are comfortable. Okay, they may be great at something, but they are also conversant using other, tools.
Right.
And that is very important. So bioinformatics, genomics, and then, climate change is real. And then, like recently I was reading a report, the temperature has gone up by 1.29 degrees centigrade or something like that. So it's, there are so many different fields now. So you just need to think what you want to do. So there is nothing like, okay, this is going out of, and that will happen only if you want to, if your focus is very narrow, that's going to happen. But then if you take a problem and if you're trying to approach it in different ways, I don't think that is going to be a problem at all. Yeah.
So talking about approaches, focus, and about scholars and students, how they handle things. What do you think, how should a scholar handle long periods of experimental failures or unsuccessful events?
So do it as a group, if it's possible. So I will give you the example, a friend of mine, 2 years, was not able to express a protein, an insect protein in E. coli. And I was able to, he gave me these, sent me his thesis once, when it was time for the submission, and I was going through the work, and there was one full page about 25 to 30 different approaches to express the protein.
Okay.
And that work alone took him about 2 years just to express the protein. Nothing worked. And once the protein was expressed, he was able to then finish his work within a year.
All right.
Okay. But unfortunately, what happened was he was working alone because there was nobody else to help him, and there was probably, different conditions there. So it could have been different if they'd been working as a group. So this is where, I will give you another example. So we were working on a certain project, and our intention was to identify the signaling molecule in phosphate starvation. Okay, how do plants sense phosphate starvation? Okay, and so we had some inclination that, okay, sespex domain proteins, they sort of have, they work like sensors. We knew that. And then we sort of knew like what the molecules could be, inositol or part of phosphates. But then what were the genes that were involved in this? And then when we were sort of working on it, there was a paper on bioRxiv who had come to the same conclusions, and they were even working on the same genes, and they had the same phenotype.
Oh.
Now, the problem was we were going to get scooped, right? So, and when we looked at the data, they almost had, they were ready to publish any moment. So what we did was we put 5 people together. So it was just me and my boss. We were working on the paper. Okay, so I was doing the analysis, and then I stopped all my work that I was doing on tomato at that time. So I said, okay, let's take a couple of weeks off. Let's work on the paper. So it was just me and my boss working on the paper. And then there were 4 people working on the experiments.
Oh,
simultaneously. Wow. So we finished everything, and then we got the paper ready in about roughly a month.
Oh, wow.
And we had it sort of communicated, and luckily, we were able to publish, both the groups in Europe, one group in Europe and Michael Hawthorne's group, and our group in Shanghai. We were able to publish the papers, one in eLife, they published in eLife, and we had it under review in Nature Plants, but then because they were going to publish it, we pulled it back, and then we fast track publishing in Molecular Plant.
Okay.
So this is so when you're working as a group, it's better. Okay.
So did you beat them to it?
No, we both published it at the same time.
Oh, okay.
So nobody got scooped there. And we were also in touch. We were also in touch with the group. And but so yeah, that was fortunate, because there was another paper we were working on, and then that one we got scooped. Okay. Because that one we were just starting off, and it was already, it was that particular work was published in Science from the US. But when that paper came out, we were, our work was pretty much preliminary. So okay. We had to publish in a different journal. So yeah, so you see that, like, when you're working, don't try to work alone, because that is the key thing, like everybody is looking for. See, when you go into a group, into a research laboratory, the key thing is teamwork. Because if you're not able to get into a teamwork kind of a mode, then it's going to be very difficult, because that is how you sort of, accomplish and solve problems which, very difficult.
Right.
Yeah.
So talking about collaboration and working together, what qualities and concerns should we look out for when we start professional collaborations?
So one thing is, like the creating a trust I think is very important. So rather than, expecting the other person to be very open, and you need to show off as a person who is very sincere. Okay, so the minute people get the idea that you're a very sincere person, then you don't need to send any invitation. Okay, people, they will just come to you. So when it's like this, and we have a saying that when the flower blooms, it need not send any invitation to the bees come on their own. So when you have a clear idea, a clear vision of what you want to do, you will automatically, sort of attract people. And in the conversation, the ensuing conversation that you try to have with your potential collaborators, they can sense the earnestness in the way you present yourself. So that way, sort of, and once they understand that, they know that, okay, if I'm collaborating with this person, I can expect only the best. So you need to get yourself to that particular point. And that is by, focusing on your work more, understanding your work more, and, like creating a clear path to what you want to do.
So when you hire a researcher or a fellow, what qualities do you look for other than academic qualities?
See, like you said, teamwork is important, and how you approach a certain topic. Okay, like if you have a failure, how do you approach it? So these are certain things. And then do you understand how well you can work in a certain environment, where your weaknesses are, your strengths are? You should be able to sort of self-critique yourself, understand it, and then how can you think about coming out of it? And also particularly how you're able to apply yourself. Okay, so that is more important. So there was one instance where we were screening a few applications, and I was screening a few, and there were multiple candidates, and one of the candidates which did not strike the other recruiters, but that affected me, was a student who gave a very good, who had no publications. But the way he approached and the cover letter and his CV and what he had written in the cover letter, it sort of touched me because it looked like he had a good idea of what he was doing and what he was trying to do if he were to be given a position in our lab. So I selected, but the others could not see it. But then when during the interview, we saw that he had the best interview. So clarity of thought is very, very important.
Right.
So that is me, but publications are also important. But in certain cases, yes, because that shows your credentials. Because when you have a paper, okay, you know what like everything is there, and you know what your contribution is to the paper. So it gives you the PIs an idea of what you are able to do. And then during the interview, you talk with them, you get to know more about what they know. So, but otherwise, like clarity of thought, I think that is pretty much important. Yeah.
All right. So, and we are coming to the end of this session. So, what would your words of advice be to the students and scholars who are new to the field of plant science research?
Okay. So, when you're new, I think, it's you will see that there'll be fewer people working in the field. Okay, so that should not be, because I like I mentioned before in my like in our discussion, that people try to this move out of like plant research and so on. But then once you are entering, you should understand the problems that you are going to face with plant research. One of the key things is that because publications are really important, and it sort of sets the stage for your growth. It's going to be difficult, so you need to keep that in mind, and then you need to sort of plan accordingly in whatever you do. So one of the things that we do in our lab in other places is just every time we run an experiment, we have a clear idea, and then every time we finish an experiment, we think about the experiment in terms of how a reviewer would think. Okay, what if the reviewer asks us this question? So can we answer it? Oh, if we can answer it, okay, let's not do that. But if you think it's not going to be a problem, if it's going to be a problem and we have to answer, then we sort of do the.
You kind of reverse engineer it.
It's sort of, you need to think, because that is how important it is, like, see, publication is not about doing your experiments properly and then publishing it. It works for at a certain level. But then if you want to, when you're creating some impactful studies and you're pushing it to higher journals, it is not about how you do the experiments alone. It's also about how you are going to sell it. That unfortunately is the case. You need to put it, okay, so you will see a lot of papers coming out, okay, this is going to transform agriculture and so on. So you sort of, like it's also about presentation. So that is very important. So these are certain aspects. So when you're getting into this, it's not that you're just going to run an experiment. That does not make you a scientist. Take it to the end, publish it. The next thing is communication is something that we lack. But then if you're able to do that, then be like sort of the icing on the cake. Yeah.
Wow, so wonderful talking to you.
Yeah, sure. So are there going to be any questions from anybody else? I think
yes. Shivu is working on that.
One question I just collected, that how to improve the quality, like during the publications, that the image quality. So the question is on the resolutions of graphs made from the R softwares. So is this any code for the improved resolutions, or it is the another way to improve it?
So we have certain image manipulating software. So like you have a certain raster graphics, or you have vector graphics. So you can use Adobe Photoshop, you have Adobe Illustrator and so on, but those are the paid versions. The open source versions are GIMP. GIMP is similar to Adobe Photoshop. And then you have not Illustrator, is Inkscape. So Inkscape is for vector graphics, and that is also free. So when you sort of prepare your figures, you make sure that you use these softwares. Okay, so one thing. Secondly is like your publication is based on figures. So your figure tells the story, like figure 1, figure 2, figure 3, and so on. Okay, and more often than not, figure 1 is going to be a composite of different figures. So we have different panels, A, B, C, D, E, and so on. So that one figure should say a story. Okay, so when you have a reader, when you have a person reading your article, you look at the figure, and just the figure alone should give you most of the information that you need from the paper. You need extra details, you go to the text. So the key thing is, okay, what is figure 1 going to be? What is it? What story is it going to tell the audience or the readers? Then figure 2, what is it going to be? What are the ones that are going to will go into this particular field? So one of the things that we do is like, okay, we create this thing right up front, and then we have panels. Okay, figure 1, okay, this, figure 2, like, okay, pipeline, and figure 3, each 2 hybrid. Okay, you leave it blank if the experiment is not finished. Okay, but you fill the other parts. And then the figure 1D is going to be something else, like, for example, say some kind of interaction, like a biomolecular fluorescence or something like that. And then, okay, that is done. So you put the figure there. And okay, something else here is missing. You leave it blank, but then you know. So you know how you're building the story. So writing a paper doesn't start after you finish your experiments. Your paper starts right when you design your experiment. The minute you start thinking, okay, you start thinking about the project, your paper starts. Yeah. So figures, yes, you can use GIMP, Illustrator. I mean, the paid versions or Inkscape for vector graphics. And also you set the figures to the page size. You can, so usually, for example, around, I think the width is going to be about 6.7 inches. The length is going to be about 8.7, 8.8 inches. So each figure, okay, I want like 4 figures on the top. So that'll going to be, okay, 2 inches by 3 inches, like 2 by 3, and so on like that. Okay, you decide the size of your figure. And then you generate the figure to that level. And then you put it on, arrange it on your, image manipulation software. Create a canvas of that size, 6.7 by 8.8 or something like that, or whatever the recommendation is from the journal. And then you start putting in the figures there.
Yes.
This is how you sort of build it. Yeah.
Okay. So let's talk to guest in Zoom. Mukul from Bangladesh and Dipti. Yes, Dipti. So I request first, Dipti, what is your questions? And from our end, I wanted to know that why you need to learn R. What is the applications of R in your research or your life? Are you there?
Yes, sir. Hello, sir.
Yeah, hi. Hi, Dipti.
Hi, sir. Sir, actually, I'm a PhD student from Karnataka.
Sure.
Like, we are working on the gene expression analysis, sir, like differentially expressed genes in the different inflammatory conditions. So I'm a beginner with the R, sir. So I need to learn the R for different plots that what you have showed just now. I'm in the learning process, sir. And we want, like, we have 3 different conditions from the inflammation, and we have collected the data sets for all the 3 conditions from the GEO database.
Okay.
Like, we need a 3D matrix comparing all the genes against all the conditions. So I just want to know whether we can use R to do this, sir. Like, if we can construct a 3D matrix using the R for comparison of the genes.
Yeah, you should be able to do it, but then I think it's I'm not in a position to like explain it to you like right away right now, but then you can sort of email me the details, and then I can sort of give you a way of how to approach it.
Yeah, sure, sir. So your mail ID, sir, can you please share the mail ID?
So I think it can be shared, right, Shubho? I think you're muted. Shubho, you're muted. Yeah.
Dipti, please mail us for requesting with email address. I will
Yes,
send
sir.
all the
Sure, sir.
informations. And from our website, tomorrow morning, you got the all information to the speakers, but it's the social media and all. So you can email
Okay,
me
sir.
for the request. I will forward the email address.
Sure,
Uh,
sir. Sure.
Thanks.
Thank
Hope
you, sir. Thank you.
hope this webinar or sessions will motivate you more on to work in our.
Yes, sir. It was very informative, sir. Like beginners like us, it was very informative.
Yes, but yes, we are discussing in that because it's 3-4 days sessions we are
Yeah, yes,
compiled in
sir.
the 2 years, so it's a little bit challenging when those are very new. But
Yes, sir.
yes, sure, that is the purpose for us to give some uh, initial starts.
Yes,
It is
yes, sir.
recorded and they can easily see it.
Yeah.
Mukul, are you there? What is your questions and why you were interested to learn R? Hello, Mukul. Yes, please.
Yes, sir. Assalamu alaikum, sir.
Salam.
Salam, Malikum salam.
At first, I would like to thank you, sir, for organizing this wonderful session. And then I am thanking the presenter. Very beautiful and informative session and lecture delivered. Sir, I asked one question that agricultural related data like completely randomized design, randomized complete block design, split plot design. I want to learn about that, and I request you in future, please organize like this session for analyzing this type experimental data. And some other software like Origin Pro, Excel Stat, we want to learn about this software. And finally, I again thank you, sir. I learned some information from this session. Thank you, sir.
Okay. So basically, what I understand, Mukul wanted to learn some plots.
Yes,
Plots
yes.
and graphs. So is there any tools or any code or script are available specifically for those those plots?
Okay, so the thing is, like, the idea, like when the today's, the tutorial, the workshop, it's what we have presented is the basic things that you need to learn to use R. Once you have this under command, once you've learned this, you don't need anybody to teach you what to do. Like I said, once you sort of want to do a certain kind of analysis, all you need to do is just Google it. Okay, I want to discover like an RBD. Okay, I want to do an RBD in R. What is it? And you will get the code. And once you look at the code, you will understand what to do with it. So you don't need anybody to teach you or anybody to train you on that. So basically, what you all need to do is just use this stuff that has been provided to you. Just work on it and then build yourself. Like once you've understood the key aspects of R, and then it's easy for you to do things on your own. You don't have to, approach anybody else for doing the analysis. So that is the intention of this particular, workshop.
Hope you got your answer, Mukul. Now,
Yes, sir.
Suvarna Sharma, why you were interested in R? Why, what is the necessity in your life for
Yes.
understanding that?
Thank you, Subha, and thank you, Dr. Satish, for the
Thank
time.
you, sir. Thank you. Thank you all, sir.
Yeah. Can I proceed?
Yes, yes. Okay.
Yeah, I'm also the grad student and I'm using R for field research data. I'm not new to R, but today's session was not only informative, but also encouraging for me. As Dr. Satish shared his life events, and I believe for every graduate student, it would be great hearing you. And my query is about the repeated measure and the mixed model. Like there are signs of autocorrelation when we do the repeated measure of the data using UAV or manual measurement. So I was trying to get idea like if there are repeated measure, and is there some way in the R that can analyze the growth curve? Like if I have the multiple data taken a repeated time series and I want to calculate the growth curve and have the parameters extracted out of it. Do you have some suggestion in this regard?
Okay, so the thing is, like, I've not performed such calculations before, like analysis before, so I'm not in a position to answer those questions for you. But then, if you really want to explore it, then maybe, like, again, you can get the email ID from Shuvo, and then maybe you can reach out to me, and maybe we can continue the discussion offline, like on email or somewhere else.
Sure, so
Yeah,
Dr.
because
Santosh,
the thing is, like, because since I need to take a look at what exactly you have and what the kind of analysis that you want to do, and then maybe I need to sort of take a look at the analysis procedures, and then maybe I'll be able to give you an idea of what can be done.
Yeah, thank you, Dr. Satish. And my
Sure.
question was also about the resolution, and Dr. Shu already mentioned you about the resolution question. Maybe once I get the email, I'll shoot out to you an email and request your help.
Oh, sure. Yeah, definitely.
Thank you.
Sure.
Thanks for.
So Dipti and Suvarna need the email. So just wait. I'm share here personally in the Zoom, and in the time being, no more questions are collected from Zoom, so YouTube. So I think it's now we are at the end, so we can sum up. For my personal point of view, I just wanted to know a few peoples are when we are try to emphasize the R's, then someone asks us to that others programming like GraphPad prisms and all, those are very attractive and easy in that way, because in R it's something, it's not visual at the first time, but that is actually that is the beauty of the art. You can calculate huge number of data at a time, but for these softwares it's very hard to calculate such type of data. So my point is that why someone using R when it has some others alternative. So because what is extra in the R? So if you just I know it's a very silly questions, but it is the initial questions of many students. Why should learn R? So it's just an application of R. If you give us some idea, not only today's talk, is little bit what R software, what can achieve, or what is the applications overall. So it will the last talk from you, last address from today's webinar.
So first and foremost, it is free.
Yes.
So when I started teaching R, the reason I wanted to teach R in IRI was because people were using like SAS, SPSS, and so on. They were all proprietary software. As a student, you don't have access to proprietary software, and you have to pay a lot of money. And again, it's like a black box, like you don't have a lot of leeway into changing a lot of parameters like you would in R. Okay, so yes, the initial learning curve is going to be difficult. But once you set in, once you understand what you're doing with it, once you understand how R works, then you're not going to, you're not going to like sort of
use anything else. Yes.
Yes, this I've seen in many of the students that attended my fellow so I've when I was a student at IRI, I used to go to different departments like divisions like physiology. I started with biotechnology, then agricultural extension, genetics, and so on, in different departments. So everywhere, I've seen people picking up R for the first time, but then going on to make it their main analysis software. Okay. The next thing is a huge user base. So which means you have a problem, there are so many people who can solve your problem. It's already out there, like more than close to, I think, 20,000 packages now. I lost count, I don't check the number of packages that are coming out in R anymore. So that's the number of packages you have. The other thing is, like, for instance, if you're thinking about bioinformatics, you have the bioconductor project. Okay. So for example, you want to do a differential expression analysis. And then, like with the seek, it's all in R, right? Okay. You have several such software. So, and that bioconductor project alone has a lot of different packages, thousands of packages there. So the what you have is quite a lot. And then the other thing is that, you don't really need to understand programming basics, so just a little bit. Okay, just one line of code, and then you can, get a powerful analysis done. And along with the flexibility that it gives you, like you saw me, like I have the code, but now when I want to make a code and a mirrored plot, I don't have to go anywhere else. I have the script with me. I just need to change it. The data set is going to change. A few things are going to change. I can all work on, within that script itself. Okay. So this is the flexibility it sort of gives us.
Okay. Yeah. Thanks.
Sure.
Hope in future we for the demand of the people and interactions of the people, we should bring you or another sessions or another topic, but it's a good start. It's a good start.
Yeah, like you said, this is the first time you're doing a workshop, but maybe I think we have learned a little bit and how to reach out to people, and maybe when we do a similar session next time around, we can plan it even better.
Even that way, or we can ask many more people to Zoom, because sometimes they are asking for the clarity of the screen, because the speed is varied in the YouTube time to time, depend the strength of the signal actually.
Yeah, definitely. So, like if you were to sort of do I'm just putting it out there, that if you were to do a session on RNA-seq analysis, then, you will have a lot of questions, right from experimental design to final analysis and the output. You can have a lot of different questions. And yeah, like you said, if you have a lot of people around, and it makes for some very good discussion, which will be useful for everyone, and particularly for researchers.
In such cases, we can also try to collaborate with your institute if their instrument and their facilities or broadcasting facilities are more, I think, more better or more capacity, because in our Zoom there is not a lot of space up there. So where we also can connect.
We can explore that. That's a possibility. Yes, we can explore more.
Because it should maintain to students should try to learn or they at least give some answers. Yes.
So today I just I'm just
yes,
Python, anything is fine.
Yes. Okay. So thank you for
having me.
Yes, it's a wonderful. Shoma, it's the end of session. Please.
But just tip of the iceberg.
Yes, it's tip of the iceberg, and nowadays it's a mandatory likewise, because it's have to know R or Python is everywhere. People are asking in interview also. You have to know. You have to know. And
learning a language, programming language, is very important. No matter what you're doing.
Yes.
Everybody has to learn at least one programming language. That is very important.
Yes. And thanks for your coming to this platform, and it's really interesting, and I circulate all the materials to those are interested, and many people are connecting through the feedback link also. So I'm going to circulate them, and if there are anyone personally email me for your contacts, I will
sure.
next one.
Yeah.
Thanks.
Thank
Thanks
you.
everyone. Please, we will see in our next webinar in this month also. So please make this community or make this family, Bioingene family, more bigger and bigger. And yes, yeah, lots of appreciation, and we are thankful. We're very happy for that. Okay, Shoma, please end the